With their bright colors and loud, far-reaching calls, red and green macaws are impossible to ignore in the rainforest, but surprisingly difficult to count. Today we will look at different methods used to estimate their populations. Estimating the population size of red and green macaws is especially challenging in tropical environments. The biggest issue is simply detecting the birds in the first place. They live in dense habitats, move frequently, and have a wide, scattered range. Because of this, every method tends to miss some individuals, making it difficult to get a reliable population estimate. As we evaluate different estimation methods, our criteria will focus on effectiveness, practicality, and safety of the birds. The first method we will look at is passive acoustic monitoring. This technique uses recording devices to capture macaw calls over time, which are analyzed to determine presence and estimate population size. It's useful because it works in most conditions, collects large amounts of data, and recordings can be reviewed multiple times. However, macaws are highly mobile and live in groups, making them hard to track and distinguish, and detection errors can occur.
The second method on our list is point counts. This involves sending an observer out to a certain spot in the forest where they'll stand for 6 to 10 minutes and record every bird they see or hear. The pros to this is that it's relatively simple to carry out since very little equipment is needed. It is also safe and stress-free for the birds. The con is low detectability. The rainforests have very dense vegetation which makes it hard to see the birds, especially when counts are done at dawn when birds are less mobile. It can also be difficult to hear the birds for the same reasons listed in the acoustics method. Our third method is our recommended method for estimating population density. It's called non-invasive genetic tagging. How this works is scientists will go out to clay licks, which is where huge flocks of parrots gather, and they'll collect any feathers that have fallen onto the ground. The DNA will be extracted from the feathers and used to ID the individuals, and this DNA will be used as a tag for mark recapture when the scientists eventually go out to collect another batch of feathers. The pros are that this is safe for birds and it's a relatively easy process for researchers. The cons are that there's bias towards detecting males. Males are at these clay licks more often than the females or juveniles who are spending more time in the nests. So again, it's that issue of not detecting the whole population or not getting the whole picture. Before committing to genetic tagging as our best method, we do have a few adjustments to make. Instead of collecting feathers at only clay licks, we recommend collecting them off of transects, which will be placed through stratified sampling. We want to stratify so we collect from locations with things like different water sources, areas with nest sites, roost sites, different elevations, and this will mean we get feathers from the whole population and not just the individuals who visit the clay licks.
Thank you for taking the time to watch our video. If you have any questions, please feel free to leave them in the comments section below and we'll get back to you shortly.